The overall goal of this procedure is to generate CD1D-based artificial antigen-presenting cells, or aAPC, for the expansion of NKT cells. This is accomplished by first adding proteins to functionalize the magnetic beads. In the second step, the initial NKT cell population from the peripheral blood is enriched. Next, the enriched T cells are co-cultured with the aAPC. In the final step, the phenotype and function of aAPC cell population are assessed. Ultimately, the purity of the expanded T cell subsets can be determined by flow cytometric analysis.
The main advantage of aAPCs over current methods that utilize dendritic cells is that there can be high donor variability with dendritic cells. aAPCs provide a reproducible, off-the-shelf, and standardized method for the expansion of NKT cells.
We first had the idea for this method when I was working with Dr. Schneck and Oki at Johns Hopkins. They have pioneered the field of using artificial antigen-presenting complexes for the expansion of classical CD8 T cell subsets. Demonstrating the procedure will be Dr. James East, a postdoctoral fellow, and Dr. Wenji Sun, a research associate from my laboratory.
Begin by transferring 1 milliliter of Dynabead M450 epoxy beads into a 5 milliliter clear borosilicate glass threaded vial. Then, rinse the beads with 3 milliliters of sterile borate buffer. In a separate 1.5 milliliter micro